Hello, hello. Welcome. Hi. Welcome to today's seminar. Thank you so much for being here today. My name is Vivian Richter. I have the privilege of heading up the marketing and communications team at the Garvan Institute, and it's wonderful to see so many familiar faces in the room. Now, before we begin, I would like to start by acknowledging the traditional custodians of the land on which we meet, the Gadigal and Bidjigal people of the Eora Nation, and I would like to pay my respects to Indigenous elders past and present. Now, our public seminar series is a really fantastic way to keep up to date with the very latest that's happening in Garvan's research, and today we have a real treat for you. We will meet DNA detectives of sorts that are working night and day to help diagnose and treat what we refer to as inborn errors of immunity. These are rare genetic immune conditions, which are rare but are also, of which there are more than 500. Now, these are incredibly difficult to diagnose, which means that patients can often face years of mysterious symptoms, recurring symptoms, and illness. Now, based at Garvan, the Clinical Immunogenomics Research Consortium Australasia, which is a bit of a mouthful, so I will refer to it as ZIRCA from here on in, is a network of expert clinicians, researchers, and geneticists that are leveraging the latest technologies to understand the cellular mechanisms that are driving these rare genetic immune conditions. At this seminar, you will hear from Professor Stuart Tangye, who is the chair and co-founder of ZIRCA, and you will also hear from Dr. Shruti Swamy, an allergist and paediatric immunologist, who will present on a nationwide study on the impact of genetic diagnosis on patients' treatment and outcomes. And we are also just incredibly fortunate to have with us today Nico Roux, whose daughter Stella was diagnosed with a rare genetic immune condition at just 18 months of age through the ZIRCA program, and who will share his personal experience of what a potential diagnosis and treatment can mean for a family. Thank you so much, Nico, for joining us today. So that just leaves me to introduce Professor Stuart Tangye, our first speaker for the day. So Stu has more than 25 years experience in the field of human cellular and molecular immunology. He is a lab head at Garvan, and he is internationally renowned for his work in discovering, diagnosing, understanding, and enabling treatments of rare immune conditions. He is actually also one of our most highly cited and highly awarded researchers. He has published more than a staggering 250 scientific papers, and he is the recipient of numerous prestigious awards, including, let me just get this right, the Premier's Prize for Excellence in Medical Biological Sciences, the prestigious Eureka Prize for Scientific Research, the Gottschalk Medal from the Australian Academy of Sciences. I could go on, but I think we would be here forever, so I will just hand over to Stu. Thank you so much.
Good morning everybody. Thanks so much for coming. Hopefully you can hear me well up the back there. Terrific. Thanks, Viv, for that very generous introduction. It's always a bit humbling to hear someone talk about yourself like that, when really it's a team effort, as you'll see what I'm going to go through today. Thanks everyone for coming. It's terrific that the Garvan and Garvan Research Foundation open the doors to the public and run these public seminars and tours and programs and so on to really allow the people who are our core supporters, or just people in the public, people on the street, who really want to get a glimpse of what we're doing. Science and medical research are really a long haul journey, as you would know. So it does take a little while to get things moving along, or to get things to a point where you have made a discovery or got a new finding, but everything we do is exciting because research is really the unknown. And I remember when I was young, talking to one of my science teachers, I think, saying, how do you know what to research? How do you know what's known? And I still ask myself that, how do we know what isn't exactly known? And this day and age of huge developments and huge advances in technologies, information technology, science, disseminating information, it's really hard to keep up with what is known and what is not known. But that's also the excitement of working in something which we hope we can push forward and help people with our science, medicine, and research outcomes. Okay, so as we've said, I'm a lab head here. I've been at Garvan for nearly 20 years, actually. It'll be 20 years next April. So for all of that time, I've been studying the immune system, particularly the human immune system. And that's what we're going to drill into today. I just want to make sure I can, whoops, that's buzzes. That's cool. Maybe I won't do that. Okay, so the immune system, you probably all know what the immune system is. Sorry, Viv, do I have a pointer? Does that just, oh, that moves my slides forward and back. Oh well, we'll wing it. Never mind. I work on the immune system, and the immune system is really important because it basically is our defense system against infection. And as you can see in that picture there, it's sort of this force field, which when it works really well. And it keeps all of these pathogens or microbes at bay and stops getting into our body and causing severe disease, from an irritating infection to something like sepsis, which can be lethal. The immune system is also really important for vaccination, because essentially vaccination, as you know, mimics what our natural infection does. It exposes the body to something we don't really want to see, like a virus or a bacteria, and it tricks the immune system into priming itself and building that armed forces, so that when we do see that real natural microbe or pathogen, the body knows what to do. And it's also very important in cancer surveillance. And again, you may know about the blockbuster drug in cancer treatment these days, well, not every, but for quite a few cancers, is what's called immunotherapy. And that's because the immune system functions to kill off some cancer cells. And sometimes it doesn't work so well. So the treatments today are really revving up the immune system to get back into being very good at cancer surveillance or tumour immunotherapy. And the way this works is in our bloodstream. The immune system is everywhere, frankly, almost everywhere. It's not a sedentary system, so we have our blood vessels, blood flows through our blood circulatory system, and in our blood there's all sorts of really cool cell types which are part of the immune system. So we've got red blood cells, which obviously are important for carrying oxygen around, but we also have the white blood cells, which is all these other cell types that I'm showing over there, which have a specialised function to fight off different types of infections. So I'm talking like bacteria, viruses, fungal infections, so yeasts and molds, but also parasites. And I like to compare our immune system, because I'm a child of the 70s, to Star Wars. So you can imagine that the stormtroopers are very much a bit like those pathogens. They're not very nice. They copy themselves really a lot. You've got they can have what's they make copies of themselves, so that they develop a lot, affect you, be really problematic, whether it's a virus or a bacteria. On the other side, of course, is the good guys, Luke Skywalker, Han Solo, Princess Leia, Chewbacca, and so on. That's our immune system, and that's what's geared with these powerful cell types, different types of cells in our blood, which does the right thing. And when those two meet, this epic battle ensues, and that's where our immune system is sort of fighting off those clonal enemies of the microbial world. And on a good day, all those invaders are destroyed. So the immune system comes out on top, we remain healthy, we clear the pathogens, and we're well. That's a little bit how what the immune system does. But sometimes the force of our immune system is not strong enough, and that can result in all sorts of diseases. And I'm a little bit biased, but I think almost every disease that we can be affected by is got some sort of contribution by a faulty immune system. Not everyone, but a lot are. And there's lots of diseases which you've probably heard of, which you may not appreciate, are driven by defects in the immune system. So what am I talking about? So some of these diseases are things like lots of infections. People can end up in hospital with severe recurrent life-threatening infections. These can be caused by genetic factors, which is what I'm going to talk about soon, but also acquired immune deficiencies. And the best example there, of course, is HIV infection, which causes AIDS, with AIDS standing for acquired immune deficiency. Cancer, not just cancer as I talked about before, where the immune system kills off cancer cells, but you can have cancer of immune cells. So you would have heard of leukemias, lymphoma, maybe myeloma. There's a whole range of autoimmune diseases. There's over 100 different types of autoimmune diseases. Some are listed there: multiple sclerosis, lupus, arthritis, where the immune system starts attacking different parts of your body. It might be all over your body, or it might be very specific to one organ, like diabetes, for example, only affects the pancreas, generally speaking. Or there's things like asthma, allergy, eczema, dermatitis. And you'll hear from Nico a little bit about that in the context of Stella's journey. So how can these diseases happen? Well, Some of them happen because we get exposed to chemicals, we get exposed to different types of toxins in the environment. It can be due to lifestyles and so on. But what I'm going to talk about today and what I'm interested in is our genes, is our genome, and how errors or defects or mistakes in our DNA can cause these types of immune-related diseases. So just breaking it down very simply, at the human level, we're all comprised of cells, and within every cell of our body, we have chromosomes. So we have 23 sets of chromosomes, 22 pairs, and then depending on sex, either XY or XX chromosomes. And those chromosomes, which is that Y-looking, not Y, X-looking shape in the middle of the picture there, sorry, that's just made of DNA. So it's, each chromosome is a big, it's like a ball of wool. All your DNA is rolled up into chromosomes. And within that DNA is where all your genes are housed. And I'll talk a bit more about genes in a second. And within the genes, they encode proteins, and proteins is pretty much what make us what we are. But if there's a mistake in one of those genes, that can have a very much a knock-on problem by in disrupting how that protein might work. So, trivia question for you. Out of these creatures, bacteria, a chicken, a human, a flea, or rice, which one do you think has the most numbers of genes? We can't see bacteria very easily with the naked eye. We can see most of those things. Anyone have a guess? Which one has the most numbers of genes? If you said bacteria, you're not quite right. If you said humans, you're not right either. Believe it or not, there's more genes in a grain of rice than there are in a human being. My point there really is to tell you, we are made up of 21,000 genes, or thereabouts, 22,000 genes, different genes. And they make our heart beat, they make our lungs breathe, they make our brain work, they make our immune system tick. And as you can see, they're different organisms of different sizes can have different types of genes. So we've got to really understand the human genes and what's called the human genome, that's the whole package, to know what's going on. And if there's a mistake in one of these genes, it can be catastrophic. I'm going to tell you an example of this here. So this is a young boy, strapping young lad, born in 1970. He born in the southern suburbs of Sydney. Grew up pretty regular life, went to the beach, joined the surf club, played soccer, you know, rolled around in the dirt, got cuts and bruises and things like this. Had a pretty good childhood. Didn't grow up to be the big wave surfer that he dreamt of being, but is doing okay as a scientist. This young boy was born about a year later in the southern states of the US. His name is David Vedder, and you may know him as the boy in the bubble. He was born, as I said, 1971, and it was known at the time, because he had a sibling, a boy in the family before him, that he may have a disease because his brother died as well. So it was decided, for debatable reasons, to put him in a sterile plastic environment, which would prevent any bacteria or viruses penetrating so that he could live sort of in this sterile environment and not be infected, because it was known that if he got infected with anything, he was at risk of death. So he spent 12 and a half of about 12 and 3/4 years of his life in a plastic bubble. The last ones were actually developed by NASA, because they were really interested in this concept as well. Again, rightly or wrongly, it's up for the discussion, but not at this talk. But that's David Vedder. You may have heard of him as the boy in the bubble. And the only two differences between these two boys, generally speaking, was one gene. David Vedder had a defect in a single gene, and that caused a disease caused a disease called X-linked severe combined immunodeficiency. So simplistically, it looked like this. David, in his genome, he had 21 or 22,000 genes, as we all do, but one of them didn't work. And instead of his immune system working, there was one cell type that was completely absent. And because of that, all those infections that he got exposed to, instead of getting wiped out, they just copied themselves and replicated and made him incredibly unwell. And in fact, he ended up dying of a viral infection when he was looking to getting treated not long after treatment. So these are called inborn errors of immunity. So the inborn meaning they're genetic, inborn meaning you're sort of born with them, not sort of, you are born with them, as opposed to something like cancers which can be caused by mutations which occur later in your life due to exposure to chemicals, exposure to radiation, smoking, and things like this. So you're sort of born with this genetic change which can compromise your immune system. So that's not me, is it? No, good. So I'm just going to tell you a little bit about these. These result from an error in just a single gene, one of those 21,000 genes, and that's enough to affect the function of the protein that that gene encodes. And that's enough to cause disease. So in those 21,000 genes, there's a lot of backup plans. So you can have some, we all have mutations, believe it or not. You might not like to hear it. But our genome is full of mutations, but most of them don't affect anything because they're neutral or there's a backup plan. But in this case, there's no backup plan. And what happens is the immune cells are very, very severely affected, and typically they present with recurrent, ongoing infection. Of course, you're born with this gene error. It doesn't take long for the symptoms to appear. So the kids present clinically within the first 6 to 12 months of life, generally speaking. So you can have really severe infection, really bad inflammation, really bad allergy, and even develop cancer. And again, Nico will touch on this from his own perspective, own personal perspective, towards the end of the session. As Viv said, there's more than 500 of these diseases caused by 500 different genes. So while each one of these is incredibly rare, there might only be 1 or 2 in Australia, or there might even be only 1 or 2 in the world. When you add all those diseases up, you add all those 550 different types of diseases up, they become relatively common. And the estimates are, it's very hard to estimate this because it changes for culture, it changes for geographically, it changes for ancestry. But we're talking about 1 in 2,000, maybe 1 in 5,000 people are affected with one of these rare genetic diseases. And they can affect many, many systems of the body. So that's the other challenge here clinically, is because these patients might first go and see a dermatologist because they've got a bad skin rash, or they might see a neurologist because they have headaches, or they might see a gastroenterologist because they have problem going to the toilet or have inflammatory bowel disease and so on. So it's quite a clinical journey for patients to finally land where they need to, which is typically seeing an immunologist or an infectious disease expert or something like that. So how do we move this forward? How does this work? So I think we've said these genetic detectors. I think that's a really apt description for what we're trying to do here. So as I said before, 21,000 genes, that sounds a lot, right? But try to picture that as 21,000 genes, which are then actually made up themselves of 6.4 billion pieces. So imagine, and I'm going to show you a picture of this in a second, a huge jigsaw, and you're trying to put it all together, but there's 6 billion pieces to put together. We've got to find which one of those pieces is missing. And as I said, as it says on this slide here, all of us, we have these changes to our DNA. There's about 3 million of them in our genome, but a lot of them don't have any effect. They're inconsequential. So how do we sift through all that information to find out what gene causes that patient's disease? And I can't decide which is my favorite analogy for this process, so I've given you 3 to choose from. So one is this 6.4 billion piece jigsaw puzzle. So imagine doing that and you can't find that one piece, or you need to find the one piece that's missing. It's the proverbial needle in the haystack, or it's like panning for gold, where you're just there sifting, looking, hoping that you can find what you're looking for. So it's very, very tricky. Clearly very tricky. It's not staring you in the face. But it's really important to get a genetic diagnosis. And again, Shruti's going to touch on this in a minute, because once you work out what causes this disease at the genetic level, you can explain either scientifically or intellectually, or more importantly, communicate that information to the patients and their families, why that person has a disease. You can then give an actual genetic diagnosis. And again, this is really important in these rare diseases, because we're talking about those cases where clinicians are like, oh wow, I've never seen this before. And that's probably the last thing patients want to hear, because they don't want to be the only person affected by this. So giving them a genetic diagnosis then allows that person to connect with other people with that disease. They might not be in Australia, they might be overseas, but it's possible. From a scientific perspective, we can understand what those genes do. Importantly, and again, Shruti's going to cover this really elegantly soon, we can really, instead of just treating symptoms of diseases, we now have access to this armory of drugs, which you've probably heard of this phrase, precision medicine or personalized therapy. And it's not a cliche, it's real. You can really be guided by the gene that's problematic, and that can make it possible to give really specific treatments, which can be targeted, but also less complications with side effects and so on. You can also develop novel therapies, and also what's relevant, of course, is family planning. You can have if it's, as I said, it's inherited, so there can be other affected individuals in the family. There could be problems which could be circumvented into future generations. So I've been working in this space for a long time. I've been working in this space since 1998, when I was obviously much, much younger. And I've loved this field because it's so much potential, and not being a clinical doctor, just being a scientist, it's great to be able to connect with people and sort of see your work advance to the clinic, even though I don't have that direct conduit to the clinic. But it's difficult. So I was like, well, how do we do this? So in the early 2010s, Garvin was getting really into this genomic space, which is terrific. And I'd had a lot of experience in this space myself, but it's very hard for one person to do this, and I've never pretend that I can do everything myself. I'm very conscious of my limitations. So the way to do this is you need to build networks, and that's what we did. 10 years ago, we built Circa, or I should say we established Circa, and I was cognizantly aware that we needed all the experts. You need all the brains trust in the room. So that involved, obviously, adult and pediatric clinical immunologists, geneticists, immunology nurses, researchers. The list goes on of all those expertise, that expertise that you needed in the one roof to be able to pull this forward, because these are rare patients and you need a lot of expertise. And what we really wanted to do was use Circa as a platform to leverage the state-of-the-art genomics and genetics technologies that Garvin was investing in, and also the functional biology that a lot of research labs were capable of doing, particularly my own and colleagues here. So we can understand cause of disease in patients with rare or novel gene defects, develop improved treatments for these rare diseases, and then also, from a research perspective, really understand consequences of these genetic changes to enhance understanding of basically how the immune system works. So 10 years later, we've got about 300 members, and this ranges from people from all stripes, whether they be in the clinic, in the lab, in patient advocacy groups, in pathology labs, in industry partners, and so on. And these were distributed across Australia and New Zealand in many, many sites. And just to put that, try to highlight that. Now, clearly there's a lot of names and so on in this slide, so please don't try to read everything. But we have very good reach across Australia, not quite yet in the Northern Territory, but that's a real aspiration for us. But we've got clinicians and terrific clinical colleagues, and you'll hear from one in a minute, who are just so invested in these patients and their families to get things better for them. So we have this network, which is truly national and even trans-Tasman, with New Zealand being very on board here. We have terrific interactions with industry partners, but also academic partners, whether they be universities, medical research institutes, patient support groups, and other initiatives across Australia. So the way this works, it's sort of simple in its complexity, or complex in its simplicity. We really rely on clinicians really spotting those patients who have these unusual phenotypes, unusual clinical pictures. They're not just garden variety infections or eczema. They're that with a little bit of absolute uncertainty to them. So the clinicians bring the patient's information to us, and that's the Circa team sitting around the table, and we decide what to do, and we have a plan in place, and we usually end up looking at their DNA, sequencing their DNA, looking at all those 6.4 billion pieces in the puzzle. We collect their blood, we look at their blood cells, and at the moment we're getting about a 35% success rate, which is terrific. But if you were thinking that the glass is half empty, you wouldn't be wrong either, because that means about 60% of patients are not getting diagnosed. So we don't stop there. We actually have an enormous program going on, which is a research program, which involves lots of labs, lots of interactions, lots of collegiality and cooperation, and lots of different model systems that each lab can harness. So we look at blood cells, we look at DNA, we look at RNA, we look at proteins. We develop models that we need to. They might be animal models like zebrafish or mouse models. We do grow cells in culture, we grow cells in test tubes, and we try to really work out what it is. And that's a long haul investment. That can take years to come to the end, where we sit around the table again, review all the data, and then report back to the clinician, who can then feed that information forward to the patient to inform them of what we found. So that's really where our work sits, is the 35% of cases diagnostic. I'm not saying that's a low-hanging fruit, but that's pretty easy pathway. And that's great when that happens, but we're often more likely to be going around that other pathway, solving the 65% of cases. So just briefly, where we're sitting at now in the last 10 years, we do diagnose about 35% of cases, which is terrific. And some of those data, it's sort of predictable. We're finding similar mistakes in the same genes that other people have reported. We're finding new mistakes in the same genes that people have seen before. But then we've had to show how those new changes cause disease. And excitingly, we are finding new errors in new genes. So we're identifying essentially new inborn errors of immunity. And again, Shruti's been really driving one of those examples as a research student here over the last few years. And we've also been able to implement therapies, which Shruti will cover off in a minute. So this is our aspiration. At the moment, we're sitting here, 35% diagnostic rate. But by implementing a whole lot of cool tools and technologies, expertise, really talented people across Australia, we are trying to get to 70% in the next couple of years. So basically doubling our diagnostic rate for these rare conditions across Australia by pulling a lot of levers that we have that are embedded within our network in individual labs or across large collaborative networks in Australia and New Zealand. So I'm going to stop there and say thank you very much for attending, and I'm going to hand over to Shruti.
Thank you, Stu. Everyone can hear me? Excellent. Thank you. Thank you for the kind introduction and for the invitation today, both by Stu and the organisers, for allowing me to present some of our, what I think is very exciting findings in the space of genomics, and particularly in the management change that comes about from a genetic diagnosis. So Stu talked really beautifully about the journey towards getting a genetic diagnosis and how fraught that can be from a perspective of an undifferentiated patient presenting in multiple different ways that can look like multiple different things and arriving at that genetic diagnosis. And that in and of itself is an amazing and phenomenal journey that the researchers in particular, but also the clinical scientists help us with. So in a way, that is the end of the diagnostic journey, for sure. But as Stu pointed out, it's actually the beginning of the rest of the journey for the patients. And Mr. Roux will talk about this in a bit, but I will also quote some of our other patients who have kindly provided some quotes regarding their process through this diagnostic journey and the arrival at the gene and what it meant for them from there on. So, inborn errors of immunity, as Stu has mentioned, are errors that affect your immune system that result in a clinical phenotype. They can affect children of all ages, as we could probably guess, but interestingly enough, we've also noticed that they do affect older people and right into adulthood. Sometimes it's just because some of these have not been diagnosed at a younger age and the patients have progressed through life carrying this inborn error and just have not been diagnosed with it. And that's really important because we are now going to talk about a little bit about precision medicine and how that affects not only the young children, who are the bulk of the diagnoses that we make, but also the older population and how it can provide some real answers and treatment guidelines for them. So obviously the diagnostic journey comes with a lot of investigations, multiple investigations, sometimes to no avail, and sometimes monitoring to try and work out what's going on with the patient, trying to chase our tail, as it were. And it also comes with a lot of, and I won't say experimental medications, but what I mean is, you know, throwing a full blanket over what we think would be beneficial to the patient. And that often comes with multiple side effects, unwanted side effects, and sometimes they just don't work. And so I think that is the conundrum that we face in the clinical and research world when it comes to all inborn errors of immunity, all inborn errors, but also inborn errors of immunity. So when we do find this particular gene, what does that mean for patients? And one of my patients said, not having a genetic diagnosis for our daughter carried so much worry and uncertainty. And I think we can all sort of relate to that, whether we do have a child affected or not. You know, it is quite a traumatic process for families. And then when we do find that particular gene, as Stu pointed out, the variations of the billions of, that they've sifted through and arrived at that final conclusion, what does that mean? It means that having a genetic diagnosis as a family, we could finally move forward and start to better understand and manage her condition, which is really what Mr. Roux is going to be talking about very soon, I suspect. So really, the end of that journey is the start of the rest of the journey with multiple little stops along the way, but at least it's a more targeted journey that the families can take from here on. The meaning behind this genetic test I found fascinating, and this is one of the patients who had recently had a child diagnosed. Genetic testing was crucial to getting to the root of her immune problems and on the right treatment path. She was prescribed the medication, which is the mainstay in her management, and consequently resulted in significant clinical improvement. And as a clinician researcher, I think that's where my excitement lies, is in finding the gene, yes, and finding the novel treatment cause, or finding a targeted treatment, but ultimately arriving at the point where there's a significant clinical improvement for the patient and improved quality of life, which is basically what everyone in Stu and Circa and the community are working towards. So how does obtaining a genetic diagnosis contribute to the treatment of the patient? And as Stu pointed out, what we wanted to know, particularly in the Circa group, is at what frequency does obtaining a diagnosis lead to a significant change in the patient management compared to before they were diagnosed? And that's a real question, because I think it's really important for us to understand and advocate for the crucialness, I guess, of finding this gene, because it helps determine a change in management from something that wasn't patient-focused or patient-centered and potentially not working or working suboptimally to something that would actually provide a meaningful intervention for the patient. So there began our journey, and we thought, right, so we have Australia, and we've got, as Stu said, members from different centres all across Australia. And really the whole thing about rare diseases, I think, is collaboration, is bringing people together from all walks of life and all sort of states of Australia, if we're talking locally, to contribute our awareness of each other's patients, so we can no longer standing with one patient, we're standing with 2 or 3. And I'll talk a little bit about a quote from a patient about what that means for their family. So we put out a call to the Circa clinicians, calling all Circa clinicians, could you please, contribute, are you interested in contributing your monogenic patients, i.e. your patients with a genetic diagnosis, to our study, so that we can then understand what that means at the very sort of physical, clinical level for you, but also we can potentially use that to advocate for ongoing availability of these testing tools in the clinical and research platform. So that's what we did, and we got responses. We got over 20 clinicians, 23 to be exact, from across Australia who contributed to our study. As Stu said, we're still waiting for Northern Territory, but hopefully that's not too far away. And what were the results of us coming together? So we have 161 patients and counting. This is a live research project at the moment, unpublished data, but I thought I was excited, so I thought we should hear about it. The patients' ages range anywhere from younger than 1 year of age, so infancy, up to the age of 65. The median age is 10 years of age, which is actually a little bit older than we would have potentially anticipated in the past specifically, and about 50% between male and female split. And there's a little sort of graph looking at sort of the age range of the patients. But what I found really interesting was how old were these patients when they were actually diagnosed? Were they all picked up at a really young age and that was it, and then sort of moved up from there? But actually, if you look at sort of the age of diagnosis, yes, most of the patients are picked up when they're younger, but actually you're getting patients picked up as they're heading into their older years as well, which is really fascinating and I think has much more clinical and broader clinical implications than we once thought in terms of pediatric immunology and what that meant for our under 18 cohort. So once everyone put their hands up, we said, right, what are the questions we'd like to ask of you without making it too onerous such that they back away, or not asking enough so we don't understand, we don't get answers to the questions that we're asking. So the first question we asked was there a major management change that came as a direct result of the diagnosis? And by that we mean, did it help push for a bone marrow transplant, or did it help guide how that bone marrow transplant worked, what chemotherapeutic agents you used, and so on, because that's quite a big life-changing sort of management change? Or were they put onto some targeted precision medicine type treatment that works for that particular gene and has been reported to work in that particular gene and disease-specific context? The second question we asked was there a minor management change as a result of the diagnosis? And by that we mean a lot of these genes, as Stu pointed out, have multiple effects on multiple different body systems. So yes, we're monitoring their immune systems, but are the genes that they have identified to have have implications in other functions. So in their endocrine system, in their neurological system. What else do we know we need to be looking out for now that we know that the patient has this diagnosis? Are there specific antibiotics, vaccinations that the patients require from a perspective of knowing that they're more susceptible to certain things based on what we know about that gene? So still relatively targeted treatment, but perhaps not life-changing, as it were, from a major management change perspective. We also asked, were there any family members that were diagnosed as a result of the proband or the initial member of the family being diagnosed? And was there genetic counselling, as Stu talked about, family planning on outcome of the diagnosis? So I'll just go through this really quickly in terms of what to look out for. So the dark blue colours are your major management change. Your orange are your minor management change. The greens are both the family member diagnoses as well as the genetic counselling, and the light blue is that there were no changes that were observed after the genetic diagnosis was made. And really we found this quite fascinating because 70% of the patients who had a genetic diagnosis had a resulting major management change, which was phenomenal, and it's something that certainly blew me away. But not just that, of those who didn't have a major management change, a further 15% of them had a minor management change. So still very targeted and therapeutic, sort of individually personalised treatment, but not in the major management change. So that's 85% so far who've had a management change of some sort. 6% had other family members diagnosed or had real implications in terms of their family planning, and 9% had no real changes in their management. And that's to be expected, I think, if you look at a bell curve. So when we look at major management change, what are we talking about here? And again, phenomenally, about 50% of patients either underwent a bone marrow transplant and or had a targeted treatment that was changed to the gene that they had been identified to have. 5%, I think this is actually really important, and we don't think about this often enough, I think, in clinical medicine, is that knowing when to withdraw care, particularly when you know that the prognosis of the child is not suitable for a life that is sustainable and a quality of life. The second two patients of that 5% were on the brink of having a bone marrow transplant and were drawn back from it because of what we know of the gene that they've been diagnosed with. I think those are very important things because a lot about what we do in medicine is not just about what we do, but also about what we don't do. That's really important in clinical management. The next question I had was, well, are the patients who have a major change in management, again, are they like super young, or do they sit sort of across the age groups as well? But again, as you can see there, yes, they're quite young, the majority and the bulk of it, but really that major change results across the age groups, across the range that we have in our cohort. So what are the effects of having this genetic diagnosis? I think it really matters on being understood. And this is the quote from the patient that I was mentioning before, that meeting people with the same genetic diagnosis makes us feel more like a closer community, because as Stu was saying, you find one or two individual patients across the world, and having a genetic diagnosis combines them all together on multiple different social platforms now to allow for support for each other. On the future and what it means for the child or the patient, as well as their family for ongoing. Now when my son plans his own family in the future, said one parent, it can potentially be avoided, the illness that is, by having the spouse tested to make sure it's not passed on. That's an amazing bit of knowledge to have as you go on and plan for your own family. When it came time to think about expanding our own family, it gave us the information we needed to make informed choices and feel prepared. The diagnosis led to another family member subsequently receiving the same diagnosis, and their medical management was then able to be refined for a better outcome for them. So again, this is beyond sort of just the patient and the wider implications that it has. So speaking of the others, what we found in our data, that 16% of patients from our cohort had a family member who was diagnosed as a result of their diagnosis. And a lot of these have had targeted therapeutic treatments initiated after their subsequent diagnosis as well. And that could be siblings, parents, grandparents, and cousins. So that's kind of spanning across all the length and breadth of your immediate family, I think. And as a result, there were 33 extra individuals who received a monogenic diagnosis who up until then were undifferentiated, unmanaged, and not receiving the right type of treatment by and large, which is very big actually. And it has a measurable impact as we've talked about. And this is something that we don't think about very often, but so this parent says, I never really understood how important it was to get a diagnosis early on, but I'm so glad it did. It gave us answers. It helped us understand what our daughter needed. It could give her support from the start. And what she was talking about there is something as simple as accessing NDIS care, where a lot of these patients don't get the opportunity to do so without having access to a genetic diagnosis. And this allowed them to do that, and which allowed them to have early intervention and early initiation of some of the other management things, including physio, OT, all the things that go with having a chronic illness. So that's my talk for today. Thank you so much for giving me the opportunity to talk today, and thank you to Prof Tangye and Paul Gray, who really took the lead on this project and then handed it over to me, and for trusting me with the project, and to all the clinicians who've contributed their data tirelessly, and of course to the patients and their families, because without which we would not know as much about the immune system as we do now. Thank you.
It is just incredible to see the impact that this incredible research has had. Has everyone got a mic? Yes. There are some incredible statistics we just saw in that presentation, Shruti. Thank you so much for sharing that. Now, to bring, I guess, a bit of a personal face to one of those people in those statistics, I'd like to throw a couple of questions to you first, Nico, if that's okay. So your daughter Stella was diagnosed with a rare genetic immune condition. First of all, can you tell us a little bit about Stella? She was your first child. First daughter. That's right, first daughter. That must have been an incredibly exciting time for you.
Yeah, so when we found out that our second child was going to be a daughter, we were really excited because now we have all a boy. You know, he was keen to be a big brother, a bit of the pigeon pair, and yeah, everything was as normal and as good as it could be. So there was no anything for us to expect anything that was the road that was lying ahead for us at the time. It was all joy, and even up to the day that she was born, it was all normal and perfect.
And you first started noticing that something was wrong quite early on, around 3 weeks, I think.
Yeah, she was. I mean, like I said, she was born full term, normal, no complications or anything. But at about 3 weeks, she started to show. It started look like what we called like pediatric dermatitis or small little spots or eczema. It's like, oh, just slap on some cream. It will, it will go away. At 3 weeks, that's when we sort of started to see something happening with her.
And tell us a little bit about how things progressed from there. I know earlier you mentioned that, just seeing her sleeping now at peace is something that's quite transformative. So she was scratching, her skin was, just in an incredible state at that time.
From that 3 week, it just got really bad. Like her whole skin was in a rash, pimples all over, cracked skin. And we clearly, and it felt like the presentation you did, Dr. Shruti, that's what we went through, is you did not know what's happening. You went for various therapies from top dermatologists in terms of light therapy, methotrexate, creams, wet dressings, like steroid creams and everything. And then you would see a little bit of, oh, that seems to work. And it works for a week. And then you're back to square one. So it was, and that went on for 18 months. It was only that finally when I think it was actually Dr. Peter Hsu who brought it to Circa when she was formally diagnosed. That's where that part of the journey where you realize like you can sigh relief like, oh, okay, now we know what's happening or what's gone wrong. And then what is the next, how do you prepare or what's the next journey?
So tell us about that diagnosis. What did that mean for Stella and for her path forward?
I think for us it was again the first sign of or a sigh of relief, like, oh, okay, well, at least. And I'm a bit of a half glass or full kind of person positive. He's like, oh, we'll just quickly go through this. And later on, we were told, oh, potentially you may need to do a bone marrow transplant. And I was like, oh, I don't know. And at the time, we were being treated with quite a specialized regime of through dermatology at Marine Shore. And it was like, well, let's finish this first and see if that. And but there's still a little bit of uncertainty. But it's only when we as a family got introduced to Dr. Paul Gray here at the Sydney Children's Hospital that and really explained the situation and the complexity or the journey that's going to lie. And for me, it was always, oh, okay, we'll just quickly go through this. It will be fine. But it was actually, and I have to say, credit to Dr. Shepherd, where I made a comment and said, oh, as long as we can get her not scratching and wiggling, because she was always scratching. Her hands had to be tied. She was picking her skin, like her fine motor skills. Even till today, it was phenomenal how she managed to like just picking at her skin. And I made the comment to say, oh, if we can just stop that, that would be phenomenal. And he sat down and he said, this is a major event that we don't do it for just to improve quality of life. It is life or death. And that's where it thinks like, okay, well, this is serious. And then preparing for the journey. But everyone, like the collaboration between Circa and Sydney Children's Hospital is phenomenal. Always said as it is, what's happening exactly at what stage. And Stella went through a lot of complications through the process. But always, I think for us as parents was to just trust the doctors. They know, the specialists. Don't go into Google and Dr. Google says this and this. Dr. Google knows nothing, That's, and really as parents, absolutely rely and we can't fault it, And I know we are on the positive side of the curve, but I think the earlier on the diagnosis, although 18 months, has really made a difference where she's now.
Can you talk us through, I guess, the transformation that you would have seen following the bone marrow transplant, which is a very invasive process, but tell us, what changed?
I think once she started to, come out of and her white cells and soldiers are picking up, and because she was in hospital for 8 months in isolation, so there was a lot of development things that she would normally do through daycare that didn't happen for her. So she have and still has a little bit of delays in development and things, but not severe that she can't attend a normal school. But just seeing her, the first observation was her skin was just perfect. Even until today, her skin is very resilient. She handles suntan very well. Like her skin is. So that was probably the first observation. But actually, just seeing her, how I think through the process, how resilient she is as a little child. She is phenomenal in terms of if she's got her head on something. Even me as a dad, it's like it's going to happen. She, but that enabled her to actually then, although at a late stage in life, because I think we only did the bone marrow transplant when she was 2.5, when she got out, then start living life as a normal toddler, go to a normal primary school without having tube feeds and that she was on a tube feeding a nose tube for probably 2.5 years. So just starting to be a normal kid. And although what I really appreciate from, and I think that's everyone in the community, not just the hospital doctors or even, the daycare, is really supporting her and not letting her feel like, oh, she's got this condition, because her confidence is there. She interacts with everyone. She's very social, annoys her older brother like a young sister should. But I've said earlier to Prof Stuart that it cannot be overstated the importance of what you guys do. I'm just one patient of every, but just seeing the change of quality of life, what has happened, we will be forever grateful for that.
Oh, thank you so much for sharing. You mentioned Stella likes to annoy her brother. What else? She's 10 years old
she's
now, is that
10.
right? What else does she like to do?
She's not 10. She's quite an active little girl. She plays hockey quite competitively. She likes gymnastics and dancing, and I think, so she's quite active. She likes reading, which is like comforting to us as parents. Like, she's very diligent in her reading. And she like her friends. She's very social. She when we ever go to school, they say that she's a little helper at school, like making sure everyone's okay, making sure everyone does what is expected to do. And although she gets additional academic support through tutoring and special, it doesn't deter her in any way. She's very well loved. So I think, as I said, we are on the positive side of the journey, but she is, enjoys life. And she likes eating. I suppose that's because she wasn't a tube feed. So she loves eating a lot.
That's incredible. Thank you so much, Nico, for sharing. That's just wonderful to hear. Stu, one question to you. You mentioned the 35% diagnosis number earlier. You started Zirca in 2015. Obviously, genomics, the program, and research worldwide has come a long way. Do you ever feel like we'll get to 100%?
Yeah, that's it's a great question, Viv, obviously, but it's hard to be too definitive about that. There's incredible complexities in genetic diagnosis. So, as I said, and I'm not trying to trivialize this, but that jigsaw puzzle analogy I think is ideal. You got to find that single change, and we all have differences in our DNA which don't cause disease. So we have to really. That's when the gold panning thing comes. You got to filter through all that other stuff and work out where the piece of gold is. And then show that actually does cause a disease, and that's very hard to do. So I think I'll be very surprised if we get to 100%. What I won't be surprised about, though, is in my lifetime as a researcher in this space, so in the last 25 years, today there's 560 of these genes. In 2000, there was less than 100. And that's because of the power of genomics. So 2010, I think it was 2010 or 2011 was the first report of the use of next generation genomic sequencing to diagnose one of these cases. And since then, it's gone whoosh in terms of the numbers of diseases and the number of genes that we're finding every year to add to that tally of 550 plus. So I think we'll keep finding new types of inborn errors. Whether we'll get to 100%, it's hard to say. I think there's going to be some cases where you might need errors in 2 genes, for example, and you can only ever find one and doesn't quite explain what's going on. But I'd like to think 70% is attainable. I think that's obviously aspirational, but I think it's achievable.
Thank you. Now, Shruti, you've been collaborating with Garvin for 6 years now, and obviously there have been huge changes in the development of the technology that's supporting this work. Can you give us a bit of an insight into, from your perspective as a clinical immunologist, how the treatment has changed even within that period of time?
Similar to Stu's comment on the exponential rise in the ability for us now to detect genetic variants, that probably not as rapidly, but is almost paralleled by precision medicine, where we are beginning to look more. And this is the thing I think about research, is not only do you find a gene, but you look at the downstream effect of what that gene or which pathways it implicates, and we can potentially target those pathways specifically to make a change. And that's what we've been seeing a lot more of now, is pathway specific treatment. And so I think it's an incredibly exciting stage, because I think the genetics is racing ahead, and the targeted treatment is trying to keep up with that. And and I doubt it'll ever catch up, but I do think it gives that little bit of extra in terms of precision medicine from what's paved in front of us by the genetics guys. So
incredible. Sounds like an exciting and very active period of this research. We'll take a couple of questions from the audience now. We've got one right up there.
Thank you. I want to know if environmental factors can sort of bring on these autoimmune problems, or is it all having to be genetic?
So I'll just the question, I guess, is about sort of environmental factors. So I think a big part of the presentation was obviously around the inborn errors of immunity being genetic, but perhaps Shruti, you're also you work on allergy, so perhaps that's something that you can speak to.
Yeah, absolutely. And I think allergy is a great little example to use in that context. So I often get asked, why are allergies rising as time is going on? Is that because, you know, the beef is being fed, the cows are being fed a certain number of antibiotics and so on? And and it's a very tricky answer to give in one word. However, what it is this kind of coming together of several different factors that contribute in one individual who's potentially more genetically susceptible, not necessarily through an inborn error of immunity, but through the several genes that they've acquired and their variances that they've acquired over the generations ahead. And there's a lot of things that go into then predisposing that person to develop allergies, for example. So, for example, we know, certain things are associated with higher risk of developing allergies. We've now noticed that patients who are born here but have parents of Asian ancestry and Asian descent are more likely to develop allergies than the Caucasian counterparts. And that's got a little bit to do with epigenetics and exposure to environmental sort of normals, I guess, including pathogens and parasites. Um, the other thing is, for example, antibiotic use and the frequency of antibiotic use, particularly in younger life. Cesarean section versus normal birth, and the too clean theory. Are we just too clean as a developed country? And I don't think any of these provide the answer in and of themselves, but I think they create the perfect storm to then kind of make that individual susceptible to that. And while that's probably not as sort of clearly applicable necessarily to inborn errors of immunity, which is where the genetic component is a much larger contributor actually to the whole kind of presentation. I guess, the answer is we are organisms that live within the environment, and, so, it's a two-way street, I guess.
How about psychological stress on a child? I read that book, The Body Keeps the Score, and it points out that children who've been brought up with a lot of abuse and stress and neglect, it can really affect their physical health.
I think that is very complex for multiple reasons.
Very complex.
Exactly.
That's why I want your answer.
Thank you for that. Look, it is complex for multiple different reasons, and one of them being that we just don't have enough data. And I talk about data, and we all talk about data a lot, because I think that's where we go from anecdotal evidence to something that's translatable across sort of, you know, um, general sort of society. And it's very difficult in that context to get data, and obviously because you could never sort of really look into that in too much detail. What I will say, however, is that I think when families are in that environment where they, so for example, are in abusive families or kind of very stressful environments growing up, there are multiple different confounding factors that go into that as well. Is there a family history of another illness, for example, that contributes to that stressful environment? Does that then sort of mean that they're already predisposed to having that particular illness? And so on and so forth. So I think there's multiple different contributing factors in that regard, and it's really tricky to tease out what's what. And it I think it would be very. I don't think I could draw a correlation in that regard. All I can say is I think there's perhaps multiple different things going into that particular sort of environment that leads to a certain outcome, whatever that may be.
Thank you.
Thank you. Thank you for the question. Thank you for the answer. I'll just go to the Zoom room because we've got a couple of great questions here. How do you determine which mutations variants are pathogenic and which are benign? How do you sift through the variants to figure that out? Stu, maybe you can take that one.
In case you couldn't catch that, the question is, how do we know a particular change in a gene causes a disease versus it just having no effect? And that's really where the research comes into it. And what I mean the research is the functional studies that we do in the labs here and elsewhere that test that. So we develop a lot of systems where we can actually test the function of a particular protein that does or does not have that variant in it because of the gene defect. And essentially, it's a bit like a rheostat, you can imagine. So when you have protein X and it's operating well, it's operating at 100%. But then if it's less than, say, 50%, you might think that's problematic or disease contributing or disease causing. You can have these mutations that result in 0% function of that protein. And that can be quite serious. So it's really comes down to what we call functional genomics or functional validation. And that's often where that cartoon that I showed, where after you get the diagnosis, but then you go into this research pipeline, it's a pipeline and you've got to look at it in many different angles, many different perspectives. We sometimes use different cell types to do it. We sometimes use animal models to do it. But that's really the rigor, where the rigor is needed, because I'm always saying to myself and my team, these are people's lives we're dealing with. It's not just a research project, which it is. But at the end of the day, we are going to report back and say, that variant, it probably caused the disease, but we've got to be right, as right as we can be in a research perspective. So it's very rigorous, it's very multi-layered, and it's very, we use a lot of approaches that validate, come back to the same point from many different angles. So all the roads have to lead to Rome in that setting. So it's a lot of lab work and protein, functional testing, biochemistry, molecular biology, all that sort of stuff. But it just filters out a lot of red herrings. You get a lot of noise. There's a lot of noise, but we've got to find the gold, in the creek.
Thank you, Stu. I am conscious we're over time, but perhaps maybe time for one more question. We've had one just at the front here.
Thank you very much. You're dealing with small numbers in the total population. And this means that when you want to push your project, the decision makers who allocate the funds are going to be saying, well, really, this is small fry. We've got much bigger fish that we have to deal with. How do you go about getting the money?
Yeah, that's an amazing question, and that is a barrier that I've been up against for a long time. Slightly less these days, because I think the idea of rare diseases being important is acknowledged by people who pull the money levers. So the question was, how do you convince funders that you might be studying a really rare disease that only affects half a dozen people in Australia? If that, why is that? Why are you going to get funding for that? So the beauty of inborn errors of immunity, apart from being very focused on one individual and their families and so on, as we talked about, what these diseases tell us about is actually fundamental mechanisms of immunology. So I'll give you a real example here. The first disease I worked on, which was in the late last century, was characterized. It was a disease that only affected males, so it's called an X-linked disease. And these boys were born, they were perfectly fine until they saw this virus called Epstein-Barr virus. So if you haven't heard of Epstein-Barr virus, you would have had Epstein-Barr virus infection. EBV causes glandular fever, or infectious mononucleosis, or kissing disease, as you may call it, depending on where you're from or what generation you're from. But EBV actually causes 7 different types of cancers. So what you've got to do is, I'm not trying to say you've got to be super clever, but what I would do is pitch my grant saying, look, I'm studying a rare disease, and the main problem here is Epstein-Barr virus. And by studying these patients, it tells us what you really need to protect against Epstein-Barr virus. And if we can work that out, we could then might make a vaccine for EBV, which could prevent not just viral infection, but might help in the setting of cancer development. So you've got to have a really big vision and an outlooking view of just beyond the small number of patients you're dealing with, but think, well, what is it about these patients that can tell us a lot more? And that's where the gold dust is, realizing that it might be a couple of patients in Australia, but if they're really susceptible to Epstein-Barr virus, or they're really susceptible to tuberculosis, or really susceptible to HPV, which causes cervical cancer, you can have a story, a narrative. I don't want to say story to make it sound like it's simplified, but it's a bona fide connection to a real problem out there. And just going back to Shruti's point, a lot of these patients that we study have really bad allergic type diseases. In the disease that Stella had, one of the complications, all the dermatitis, that was all about allergy and that type of disease, eczema. Again, so we've got these models of humans who have just one genetic defect, but it totally derails the immune system. And we see that derailing in many types of conditions. Allergies rising in the general community, autoimmunity is rising in the general community. We can understand how it works at the molecular level and then hopefully intervene into the general population who has a similar clinical problem, maybe just not as life-threatening as is it in these extreme cases.
Thank you so much. That's all we've got time for today. I just want to thank you so much to everyone who's come on site for this incredible seminar, and everyone who's joined us online. Can we please give a big round of applause to all of our speakers today?